Good morning, ladies and gentlemen, and welcome to the third lecture of Chapter 8, talking about skeletal muscle. Today, we're going to try to put together everything we've discussed for the first two lectures and how that all relates to muscular contractions. And so, let's start off with a quick review. So, let's look at our sarcomere, which we had discussed in the first lecture under muscle anatomy. And so, as we know, the sarcomere is the basic contractile unit of the muscle. So this is the portion that that allows muscles to contract and produce mechanical energy or also power. This is nothing new. This is something that we're all very familiar with. Now, as a quick review, right? Within here, within the sarcomere itself, we have what's called actin filaments, aka thin filaments, and we also have myosin filaments. Also known as thick filaments. So you'll hear these terms, used interchangeably, but during this lecture, we're going to stick with actin and myosin. Now, when relating to the sarcomere skeletal muscle anatomy to movement or mechanical energy, there's this theory denoted as the sliding filament theory. And essentially what the sliding filament theory states is it's the mechanism to which muscles contract. And how this works is basically what we're saying under the sliding filament theory is that thin and thick filaments slide past each other to make the actual sarcomere itself smaller. Oops, can't spell. And with the contraction of the sarcomere itself, this allows us to produce mechanical energy. And in theory, this is how our muscles work. So, how does the sliding filament theory actually come to play? Well, when we do this, we need to identify a few key portions of muscle anatomy first. The first, of course, is, again, our actin filament. And our myosin. And on the actin filament, right, there's a couple of subcomponents. You have the actual actin itself. You have this portion called troponin. And then you have the fiber that crosses or weaves over and around the actin itself, right, called tropomyosin. And so what the tropomyosin does in itself is it blocks the myosin heads from binding to the actin. Now, how does the body get around that? Well, that happens with the release of calcium through the sarcoplasmic reticulum. So in the second lecture, when we talked about neuromuscular signaling, at the very end, we talked about how the neuromuscular signal went down the T tubules and then went through the triad, which signaled the sarcoplasmic reticulum, also denoted as SR, to release calcium into the muscle. And then, so what happens with the calcium ions that are released into the muscle itself? It'll actually bind to an active site in troponin. And then what happens when 4 calcium ions bind to troponin? It causes a conformational change which moves the tropomyosin out of the way, allowing, so it's assumed that the tropomyosin has been moved out of the way here, allowing the myosin head to bind directly to the actin. And this is how we get into the cross bridge formation, which is the first step in the sliding filament theory. And so in order for the cross bridge formation to occur, remember calcium needs to bind to troponin first to allow the active site to be unblocked in order for the myosin head to bind to the actin itself. And so once that occurs, once the calcium is released from the sarcoplasmic reticulum, binds to the troponin, and we have this conformational change moving the tropomyosin out of the way to unblock the actin, we create our cross bridge. And this gets us into step 1 of what we call the cross bridge cycle. And this is the crux of the sliding filament theory. And the cross bridge cycle contains 4 steps. Okay? And so here in our artsy little diagram that I have done my best to kind of draw out, we're going to go over the first step. So, with step 1, we have the cross bridge formation. And so what we are assuming at this point is within our actin, the calcium 2+ is already bound to the troponin, and the tropomyosin is moved out of the way. Okay? And so with this change of the tropomyosin out of the way, all we have left, and I'm just going to show from this point on, is the actin filament itself. Okay, let's clean up our board here a little bit to give us a little more room to write and explain this first step. And so what happens in the myosin head with the cross bridge formation is when the tropomyosin moves out of the way, the myosin head attaches to the actin active site itself. So what this looks like is that myosin head has moved down and is attached to the actin itself. And within the the myosin head, we have an enzyme called ATPase. And what ATPase is, it's an enzyme that hydrolyzes ATP. So what this means is ATP becomes ADP plus 1 inorganic phosphate. And what that happens is that ATP has been hydrolyzed during the resting stage during step 1, right before the cross-bridge cycle. And so what we have here within our head, so the adenosine portion is going to be red, and our phosphate groups are going to be in yellow, right? And so we have three different ATPs here. Excuse me, 3 different phosphate groups at this point. And they stay like this, hydrolyzed, but still within the myosin head during the cross-bridge formation. And as the myosin head attaches to the actin, this particular phosphate that has been hydrolyzed is released, causing this bond between the myosin and the actin to become stronger. And so this is how we get the cross-bridge formation. So let's clean this out a little bit and move on to our next step. Okay? All right. So we have our cross-bridge formed. This brings us into step 2. Which we can call the power stroke. So at this point, within step 2, let's go ahead and get rid of our relaxed state of our myosin head here. Our myosin head is attached to the actin itself. And our phosphate has been moved. And so what happens at this point, the ADP is released. And what this causes is for the myosin head to pull on the actin filament, causing the actin to go this way or towards the center of the sarcomere itself, causing the shortening of the sarcomere. And so at this point, what this all looks like is, let me clean this up here. Our myosin head, I'm going to make this green at this point, cocks itself causing that pulling on the actin filament, causing it going this way. And that's what we call the power stroke. The power stroke is essentially the mechanical movement of the actin filament causing the shortening of the sarcomere. Now, this moves us into the third step. Clean this up. Get rid of this. Step 3, which is what we call the cross-bridge detachment. So what happens is, in order to keep the process going, we have to add another ATP to our myosin head, because we have utilized our energy at this point to create the power stroke itself. And so what happens is we have an ATP that comes in and attaches itself to the myosin head portion or the ATP ACE of the myosin head. And so, again, our red is our adenosine, and these three little yellow dots are our phosphates. And so, what this happens is once this is attached, and the ATP is attached to the myosin head, it causes the myosin head to detach from the actin itself, looking like this. And so once the myosin head is detached, we can move into step 4, which is what we can call the reactivation of the myosin head. And essentially all this does is that ATPase hydrolyzes the ATP, causing this phosphate group to break off, but still stay attached to the myosin head itself, and causing the myosin head itself to move back into its relaxation state, ready to repeat the cycle itself. So essentially with step 4, you're resetting the myosin head to continue the cross bridge cycle, to continue the muscular contraction. Now, what I want to point out is this 4-step process will continue as long as calcium is available to keep the actin filaments. The one aspect I do want to point out is the cross-bridge cycle will continue to go on as long as a couple of things are still available. The first is calcium is still available to keep the tropomyosin in its conformational form to allow the actin active sites to be unblocked. Second, as long as there's ATP available for the cross-bridge cycle to continue, or the muscle's not fully contracted. So bottom line is the 4 steps of the cross-bridge cycle will continue until the muscle is fully contracted. And so what this does is the cross-bridge cycle is the mechanism that allows the sliding filament theory to occur, and how the muscles create mechanical energy from chemical energy. And that chemical energy, it comes from ATP. I hope this helps. If you have any questions, please feel free to reach out, and also follow along with the associated slides that have been posted on D2O. Take care, and I'll talk to you soon.